Plants have mitochondria that produce cellular energy in the same ways that mitochondria in animal cells do. They can metabolize glucose and use the energy stored in those bonds to make ATP using an FO-F1 ATP synthase pump. However, plants also have chloroplasts. Chloroplasts are like mitochondria in some ways, and in other ways they are very different. Both chloroplasts and mitochondria likely were free-living organisms before they became symbiotic with a precursor to modern eukaryotic cells. However, prior to symbiosis, chloroplasts were photosynthetic prokaryotes. That means they are able to convert the energy from light into a food source. Plants are autotrophs. That means they can create their own food. This happens in two stages. One is the light reactions, and two, the Calvin cycle. The chloroplasts are the location of both systems. Light reactions convert light into cellular energy, and the Calvin cycle uses that energy to make carbohydrates. Let's look at the structure of the chloroplast, and from that we can start to learn about how that structure imparts its function. Chloroplasts consist of an outer membrane and an inner membrane. The inner membrane contains a dense fluid called the stroma. Suspended within the stroma is the thylakoid system. The thylakoid membrane surrounds an internal space called the thylakoid space. From the outside, the thylakoid looks like stacks of pancakes, but they are really a long system of interconnected spaces. These disks are the location for our first set of reactions, the light reactions. The light reactions occur in the thylakoid membrane and convert light energy into cellular energy in the form of ATP and NADPH. NADPH stands for nicotinamide adenine dinucleotide phosphate, and it is the plant equivalent of NADH. The P stands for phosphate, but we can remember this by imagining that it stands for plants. NADPH is the reduced form of NADP+. Just like NAD+, it can participate in oxidation-reduction reactions. We can refer to those reactions here as redox reactions. This is an overview of the light reactions. We see that this system looks very similar to the electron transport chain, and in many ways it is. There are two main light-collecting proteins, and cytochrome B6, which pumps protons into the thylakoid space. And finally, NADP+ reductase. With a name like NADP+ reductase, it's not hard to imagine what this does. It takes those electrons that have made their way from photosystem 2 and 1 and use them to reduce NADP+ into NADPH. The ATP and NADPH that was generated by the light reactions is then transferred to the stroma, where it participates in the Calvin cycle. Our overview of the Calvin cycle reveals that it is a lot like a shorter version of the TCA cycle, only in this case, carbohydrates come out of the cycle instead of going into it. The input for this cycle is CO2. 3 CO2 molecules are energized by ATP and NADPH to make glyceraldehyde 3-phosphate, and 2 of those are combined to make 1 glucose molecule. Thus, the Calvin cycle has captured the energy from your light reactions to make sugar, which can then be used by the mitochondria to make energy, and for making structures of the plant, like stems and branches. Let's take a step-by-step look at the light reactions and then the Calvin cycle, much as we did for glycolysis in the TCA cycle. The first step of the light reactions is photosystem 2. This is because they are named for the discovery date and not their order in the pathway. Photosystem 2 has several light harvesting complexes, which surround the reaction center complex. Bound inside each light harvesting complex are pigments. These are molecules that absorb and give off light. Light is made of photons. But photons are also a little bit like a wave. They have a frequency and a wavelength. Our eyes see different wavelengths of light as different colors. The visible spectrum of light comes from the wavelengths between 380 and 750 nanometers. There is energy associated with each wavelength. The shorter the wavelength, that is, the faster the wave is moving, the more energy it has. We know that electrons orbit atoms in discrete shells. We also have seen that electrons can only absorb energy that matches the difference between two shells, like going upstairs. It is impossible to go up half a step. Pigments are molecules that can absorb light photons. However, each molecule can only absorb the specific amounts of energy that are capable of pushing its electrons into higher shells. Since light energy is related to wavelength, it is also related to color. So pigments absorb some energies or wavelengths, or colors, and not others. Tree leaves are green because the pigment molecules in the light harvesting complexes are absorbing violet, blue, and red and letting the green be reflected. That is, just bounce off. What's cool about electrons is that once they have absorbed energy and gone up to a higher shell, they can release that energy when they go back down again. In this way, the electrons of each pigment molecule become excited by the energy given off from the previous pigment's electrons going back down. The light energy is absorbed and excites a pigment. That pigment then returns to normal, the ground state, and fires that energy over to the next pigment molecule, which gets excited and then passes that energy again to the next pigment molecule. This happens until the energy reaches a special set of pigments, which are referred to as chlorophyll 680. Chlorophyll 680 is part of the reaction center complex. This complex includes two molecules of chlorophyll 680 and a primary electron acceptor, pheophytin alpha. When the electrons of chlorophyll 680 are excited, they are released. In other words, chlorophyll 680 is oxidized by light energy to chlorophyll 680+. Pheophytin alpha accepts electrons given off by chlorophyll 680. In other words, it is reduced. These electrons are then passed off to plastoquinone. The electrons that were lost by chlorophyll 680+ need to be replaced. This is accomplished by an enzyme that steals electrons from water. The water is split into 2 protons, 1 oxygen atom, and 2 electrons, which are used to replace those lost from chlorophyll 680. So water is oxidized and chlorophyll 680 is reduced. This is the key place where plants release oxygen, having taken it from water. Additionally, the protons that are released from water help form a concentration gradient of protons across the thylakoid membrane. This will be important later. Plastoquinone now takes the electrons from photosystem II and transports them to cytochrome B6. Here, more redox reactions occur and 4 protons are pumped into the thylakoid space. In mitochondria, we saw that protons were pumped out, but here they are pumped in. This contributes to the concentration gradient that started with the oxidation of water. Next door, photosystem I, in much the same way as photosystem II, is transferring light energy to chlorophyll molecules. This time it is chlorophyll 700. This light energy then oxidizes chlorophyll 700 and those electrons are passed to a primary electron acceptor and then to ferredoxin. From here, the electrons are taken by the appropriately named NADP+ reductase and added to NADP+, which is reduced to NADPH. This reaction helps the proton gradient by removing H+ from the stroma. Remember those electrons from photosystem II? The last we saw of them, they were bouncing around inside cytochrome B6. Well, now they are picked up by plastocyanin and transferred to the chlorophyll 700 in photosystem I to reduce it from chlorophyll 700+ to chlorophyll 700. Now that the photosynthetic cells have spent all this time moving electrons down an electron transport chain and creating a concentration gradient across the thylakoid membrane, they can use this to drive, you guessed it, an ATP synthase. So the light reactions use the energy from light to transfer electrons from water to NADPH, and it uses the protons released during this reaction to make ATP. That's kind of cool, huh? So, what does the plant do with all this ATP and NADPH? It makes sugars, of course. Now that we have left the thylakoid membrane and are inside the stroma, we can observe the Calvin cycle, step by step. An overview shows us that the Calvin cycle takes CO2 from the atmosphere. It does this by opening stroma, which are basically openings in the leaf that allow for CO2 to diffuse in. The plant then combines those CO2 molecules with ribulose bisphosphate and produces glucose. The things we want to remember from the Calvin cycle are the names of the products and where energy is used. The first step, which combines 3 ribulose bisphosphate molecules, with 3 CO2 molecules produces 6 molecules of 3-phosphoglycerate. This is catalyzed by the protein rubisco. The only enzymes we need to remember are rubisco. Rubisco is the most abundant protein on the entire planet, and worldwide, there are about 1,000 kilograms of rubisco made per second. In your lifetime, you will consume almost 40 kilograms of rubisco. Rubisco is resistant to temperature and pH changes, so it will continue to work long after the plant dies. It works to promote the breakdown of the plant matter. Rubisco is the giver of life, but it is also the mortician. So ATP is used to convert 3-phosphoglycerate into 1,3-bisphosphoglycerate. And then NADPH is used to convert this to glyceraldehyde 3-phosphate. One of these glyceraldehyde 3-phosphate molecules is taken away to be used to make other sugars like glucose and cytoheptulose. Finally, 3 more ATP are used up to convert the remaining 5 glyceraldehyde 3-phosphate molecules back to 3 ribulose bisphosphate molecules, which can then be put back into the cycle. The original 3 carbons from the 3 CO2 molecules have been combined into glyceraldehyde 3-phosphate, using the ATP and NADPH produced by the light reactions. So let's take a moment to reflect on what our knowledge of metabolism has shown us about the way life works. Plants can take light from the sun and convert that light into cellular energy, like ATP and NADPH. They also convert water into oxygen, which they release. The cellular energy that they have created is used to build carbohydrates. Animals breathe in the oxygen produced by plants, and they eat the carbohydrates from the plants as well. Then, they take those carbohydrates and break them down into CO2 to produce ATP and NADH. That NADH is then used by the electron transport chain to produce more cellular energy in the form of ATP. At the end of the electron transport chain, the oxygen that the animal breathed in is reduced to form water. Animals have taken the oxygen and carbohydrates from plants and turned them into water and CO2. Plants take up the water and CO2 to produce more oxygen and carbohydrates. It's not hard to see that we are all connected, not just to each other, but to all the plants and animals that make up the biosphere, and beyond, to the massive star at the center of our solar system, which provides the light energy needed for us to continue turning CO2 and water into oxygen and carbohydrates in an unbroken chain of interactions that reaches back, not just to the beginning of life, but to the beginning of the entire solar system.